What? Go.
Tuberculosis is a disease caused by Mycobacterium tuberculosis. This disease is spread through close quarters contact like coughing. And symptoms include coughing up blood, bad cough for over 3 weeks, weakness or fatigue, and no appetite, chills, or weight loss. It is normally treated with antibiotics, but in some cases there's a mutation in the M. tuberculosis where the current antibiotics cannot consume the mutated M. tuberculosis, which will lead to the research of different methods to treat the disease. This is Mycobacterium tuberculosis. The cell wall is represented by the outer rubber, and this is represents the peptidoglycan layer and the lipid bilayer. And on the inside we have candies and air, which is going to represent the cytoplasm and the organelles. Now if you can break this wall, second cut. As we break the cell wall, that's very tough to do, the cell becomes unstable and the wall breaks, exposing the cytoplasm and the organelles. This allows for the cell to be either totally annihilated or just eaten by a white blood cell or other things. And it is very delicious.
One way of killing multi-drug resistant tuberculosis is by targeting the dealanine-dealanine dipeptide bonds on the cell wall. If you inhibit this pathway, the cell wall is going to be either weakened or die. Because of this is a good target to make a drug to treat multi-drug resistant tuberculosis. Before we can understand how we can inhibit the pathway of making these dipeptides, we first have to understand what the mechanism to make this is made and how it's made. The dealanine-dealanine dipeptide bonds are made by a protein called DDL, dealanine-dealanine ligase. Notice that this protein is symmetrical, meaning that this side and this side are exactly the same. Because of this, the same bonding and sites and everything that happens on this side happens on this side as well. To simplify our explanation, we're just going to focus on one side, although keep in mind that the same thing is happening on the other side as well. DDL can make the dipeptide bond with 2 dealanines and an ATP. DDL has 2 binding sites, one for each of the dealanine molecules. Notice that our model shows that one of these has a triangle active site and the other one does not. This is to represent the fact that one of the dealanines binds very well to the DDL, it has a high affinity, whereas the second binding site has less affinity for the DDL. Another thing to notice before we start to talk about the mechanism is that in DDL's current conformation, ATP is not able to bind to the molecule. Therefore, we know that there has to be a conformational change that occurs. First, the mechanism that this reaction works is that one dealanine will bind to DDL. Next, there is a conformational change. This conformational change exposes the active site for ATP and the second dealanine. With all these molecules placed like this, the 2 dealanines can combine to form a dipeptide bond, which is used to make the cell wall of M. tuberculosis. To inhibit DDL, a molecule called DCS is used. In the presence of DCS, a dealanine molecule will bind to the DDL. Once this happens, there again is a conformational change in the dealanine-dealanine ligase. However, when DCS binds to the molecule, there is another conformational change in the dealanine-dealanine ligase. Not only does the DCS block the active site so ATP can't get in, but the conformational change does not allow this ATP to fit into the DDL. Without ATP and without the second dealanine, it can't form the dealanine-dealanine dipeptide bond, which is used to make the cell wall of the M. tuberculosis. If the cell wall can't be made, then it's going to weaken the cell, and that's going to lead to the destruction of the M. tuberculosis cells. This is a good treatment for multi-drug-resistant tuberculosis. Notice on our model that one of the active sites on DDL is sort of triangular, but has a square at the bottom. In our model, we have dealanine has a triangular active site, meaning that it could kind of bond to it, but however, DCS fits a lot better into the active site. We showed this on our model because DCS has a higher affinity for DCS has a higher affinity for DDL than dealanine does. Therefore, in the presence of DCS is always going to bind to the second active site and inhibit the production of this dipeptide, thus treating multi-drug resistant tuberculosis.